I'll introduce them both at the same time. Jerry Weiland from USDA ARS, and Wendy Hoashi-Erhardt from WSU Puyallup. We'll be talking about a survey looking at verticillium and red raspberry screening. With that, I think Jerry's starting, right? Yep. So, laser fork back. Okay, thank you. Do you want me to give you any sort of time?
Yeah, stand up at 5 or wait?
5 minutes till total?
Well, 5 minutes till 11:20.
Okay.
I'll stop at 11:20.
Okay.
So my goal is to stop around 11:20, which is a little less time than I thought. I apologize for the dark background on the slides. I didn't remember that we have the light glare, so I will talk through this. I am one of the many Oregon researchers who helps subsidize the Washington fruit industry with some of our personal research funds, and it is our pleasure to do so. I'm going to be giving the first half of the results on the survey for verticillium. A quick background. Basically, this started when many of you growers noticed that a lot of the symptoms in your fields do not look like what we think of as the typical Phytophthora symptoms, where you see the floricanes collapse and die around June when the fruit are starting to ripen. And a lot of you are seeing primocanes collapsing and dying later in summer. And that late summer symptoms is very similar to what vert does in other crops. You see verticillium causing other crops to wilt and die in late summer. And I know many of you growers have had sent out samples, and a few of you have gotten back results saying verticillium is there. So that's one of the reasons we started looking for verticillium. The second is on the lower picture, there are some stunted, off-color red raspberries there at the end of rows typically, and verticillium can cause stunting in other crops as well. So we decided, let's go ahead and do a survey for verticillium, see what we find. Just a brief update on what verticillium is. It's a soil-borne fungus. It produces these tiny little microscopic microsclerotia, which you see here. This is what lasts in the soil and infects your plants, and it can last for decades once you have it in the soil. And we've known for 100 years that verticillium can cause red raspberries to wilt and die in late summer. There's research from 100 years ago from Washington State saying that symptoms appear on primocanes in late summer, so this is not unusual. It's just been in the background all along. Okay, so our two main goals for this research is verticillium a problem for you guys? And we wanted to look at two methods of determining whether verticillium is there, because one of the methods may be more sensitive than another method in terms of determining whether verticillium is there. I'm not going to go into that this time. We just have preliminary results at this point. So we sampled soils, and a little bit about how we sampled. We went to sites that we did not think Phytophthora would be an issue. So we went to drier sites. We went to sites that are showing wilting and decline symptoms late in the summer, not earlier in the spring or in June when Phytophthora is expected to be a problem. We looked for symptoms that were different than P. rubi, so instead of the floricanes wilting and dying, we looked for primocanes wilting and dying. And as mentioned, fields without a historical P. rubi problem. Symptoms we looked for are wilting, cane dieback, sparse canopy. We sampled 23 samples in all this year. 3 of those samples were lost because of the furlough. So we came up at the end of September when we were going to process all these plant samples. The furlough happened, and I basically had nobody to work on these samples. So I had hundreds of samples in the cooler. They were sitting there rotting, and that bugged me. So I bothered my colleagues at Oregon State University. I personally plated all of these hundreds of samples over at Oregon State University, so it was not illegal. I am a courtesy faculty there, and I used my courtesy faculty status to do that. So because there's only one of me instead of 3 that are in my lab, I did not get to all the samples, but I did get to 20 of the sites. And at this point, I mentioned we are looking at both the plant and the soil at all these sites with 2 methods. There's a cultural method that's growing the verticillium out on Petri plates. The PCR method is simply detecting the DNA. Right now, all we have are the results from culturing the plants onto Petri plates and seeing what grows out. We avoided fields. Now we get to the pretty picture part. We avoided fields. This is a great field showing Phytophthora rubi. You have a long row of almost all dead plants. All of this weed mass down here, almost all of it is horsetail. That's telling me this is a very wet site. The canes that are dying, you can tell they died early in the season. They're only a foot and a half tall, or in a lot of cases, I'm not showing this, it's the flora canes that died at the fruit season. These had been dead quite a while by the time we got there in July through to September. So we avoided these sites. We did not sample from those. This is more typical of what we went to. This is the stunted off-color plants that aren't doing well. This year we focused though on the primocanes that were wilting and suddenly dying in late summer. Here's one wilting in September. We also saw this where the plants had wilted in July, recovered and started growing again. This is very typical of Verticillium. Here you have some canes, some primocanes that are off color and just not quite as thrifty looking. These are all typical symptoms of vert. It seemed reasonable to suspect that this might be vert. And these canes are about 6 to 8 feet tall. They're much taller than I am. So this is definitely summer. Go down to the base and look at the base. You see this necrosis, that's death spreading up from the roots. So whatever is coming up from the roots is pretty strong and it's pushing up into the stems. And this is what we chose to isolate from. It's important to note that this discoloration is also typical of older stems. So you'll see older stems change color, and it's not until you scrape underneath and see discoloration. So here I've scraped off the bark, and you can scrape off that outer part and not see any lesion spreading up from the base at all. It's a lot of these stems just turn dark purple at the base as they mature. But on every single one of these that had symptoms like this and the wilting, you go down to the base, you scrape off the base, and there is this dark lesion spreading up from the roots. So whatever is coming up from the roots is causing this lesion here. So that seemed a reasonable point for us to isolate from that. And that's where we typically get Verticillium in other crop species. And value-added component. Both Inga and I, Chris Benedict, Inga, and I are all collaborating on this. We decided, we wanted to give you some value added for the research money you've given to us. We wanted to see what other things were coming up and associated with these declining plants. So in addition to looking for Verticillium, we looked for what else came out of those lesions coming up from the soil. And Inga also did a nematode count from the roots that we collected from a number of those sites. And this is simply showing on these Petri plates, we can isolate Verticillium. That's Verticillium here coming out on plated raspberry stem. This is a bunch of Fusarium and Alternaria. So what did we get? Most commonly, let's start at the beginning. So we've got the number of sites. We sampled 20 sites, or I processed 20 sites. Verticillium, we only found Verticillium at one site from these lesions. That doesn't mean Verticillium is not a big problem, but I'll explain that more in a moment. 12 of those sites that we specifically selected that should not have a P. rubi, 12 of them had Phytophthora. So even on your dry sites, we are getting Phytophthora out of those lesions. That's pretty darn important. We also isolated a lot of Fusarium and Alternaria from those dying canes. And Inga got quite a bit of Pratylenchus from all of those sites. So these are what we're finding the most right now. And I'll get back to in just a moment why we might be receiving low amounts of Verticillium at this point. If we look on a plant basis, percent plants infected, we have 90 plant samples here, and because we did not have as many root samples, this percentage for Pratylenchus is from 60-ish plants. Again, Verticillium is a low percentage of those plants that we assessed. We didn't isolate it a lot. Quite a bit of Phytophthora, 25%, about 45% Fusarium from sick canes, and Alternaria was also on about a third of those canes that were dying. Pratylenchus, 70% of the 60 plants was associated with that. So the points I want to make. We didn't find a lot of verticillium. That does not mean it's not there. These are lesions coming up from the soil. It could very well be that Phytophthora rubi is what is going to outcompete verticillium in the culture method. That is why we are using the DNA method as well to assess whether verticillium is there. Phytophthora may just be a much stronger colonizer. We've already seen all the damage that Phytophthora can do to your red raspberries. And it may be if Phytophthora rubi is in your site, it is just going to outcompete verticillium in that lesion, and we're not going to isolate it. So we'll know more once we get our soil analyses done. We'll have our culture plates from soil done in a couple months here. We're still working on the DNA method. We will get that done. The other point I want to make, so yes, we have 3 other analyses yet to complete on these same analyses, or on these same samples, before we have a final determination on verticillium for 2013. Next important point, Phytophthora rubi is still hugely important, even on dry sites where you don't expect it to be a problem. And there's a consistent association of plants, sick plants now, with Alternaria, Fusarium, and Pradolanchus in addition to Phytophthora. And with that, I'm going to turn it over to Wendy.
Thanks, Jerry. In conjunction with the field verticillium survey that was going on, Jerry and I also initiated a study in the greenhouse looking at verticillium and the response of red raspberry cultivars to that pathogen. We initiated this study in 2012 out of concern that verticillium may be playing an important role in plant disease symptoms we were seeing in Whatcom County in that year. As Jerry described some of those symptoms, they did not seem characteristic of Phytophthora root rot. And so we started suspecting other pathogens. Then Jerry was digging around and was able to culture and isolate verticillium from symptomatic Saanich and Meeker plants, and that pointed to the possibility that verticillium was playing an important role in raspberry health in the region. So, lacking significant literature on verticillium in red raspberry, we wondered how do cultivars respond to this pathogen, and are there some cultivars that are more resistant or more tolerant of the disease than others? From a plant breeding perspective, if verticillium is becoming a more important pathogen, then knowing which genotypes resist the disease would be important for improving the crop in the future. So we started by collecting and propagating 36 raspberry cultivars, first in tissue culture and then putting them in the greenhouse as plugs. At the same time, we were culturing and bulking up the verticillium inoculation, or inoculant, that we would then use in the greenhouse. In May of this year, 2013, we then planted the plug plants of all 36 red raspberry cultivars in the greenhouse into sterilized sandy soil, and then inoculated half of them with verticillium. We did this with 5 replicates, so each cultivar had 10 plants. Then, maintaining the greenhouse, we tried to have temperature and light conditions that would be similar to what they would receive in the field. And we also duplicated the setup so we would have a repeated study that was running simultaneously. We monitored plant growth and looked for disease symptoms for the next 5 or 6 months. Now, as Jerry mentioned, stunting is one of the major symptoms that verticillium appears to cause in red raspberry. So plant height is one of the measurements that we're consistently monitoring, and those are the results I'm presenting today. We're going to take a look first at Munger, which we included in the study as a known susceptible black raspberry. Now, this photograph was taken late in fall, so a lot of the canes have started to lose their leaves. But what we're trying to look at here is the cane height. And on the left, we have the 5 control plants. On the right, we have the Verticillium inoculated plants. And the response is very variable, as you can see. Hopefully you can see the cane heights there. But for the control plants, each of the canes is somewhere between 2 feet and 5 feet tall. The tape marks on each stake indicates a foot of growth, or 30 centimeters. And on the Verticillium side, only 3 of the canes, 3 out of 5 canes, have reached similar heights. And it's difficult to see in this photograph, but if we take a look closer up, 2 of the 5 plants have not grown hardly at all over the 5 or 6 months and remain only a few inches tall, indicating a considerable amount of stunting in that set. A word about variability in response. It seems that often when we're doing these studies, we see a lot of variability in the response. This is a photograph from a similar study that Jerry did, albeit with a different inoculation method. On the left, he has 3 control plants, also of Munger. The yellow foliage is due to mite damage, not Verticillium. And on the right, are 3 Verticillium inoculated plants. And there is a considerable amount of stunting in the Verticillium plants, but at least one affected plant looks better in terms of growth and vigor than the controls. So going back to our Mungers, there are 2 canes. It is a bit odd that 2 of the Verticillium canes are taller than any of the control plants. But looking close up, they're also quite spindly and narrow, and I'm confident that if we were able to take the dry weights in both of these sets, we would see a greater biomass accumulation in the controls. Moving on to Meeker. These plants are a bit less variable, which is good, which is what I like to see. The trend here is clear that the Verticillium affected plants are shorter and are showing more stunting than the control plants. Schimanus is showing a similar story, and here we're starting also to see the effect of cultivar, meaning that different cultivars are naturally shorter or grow more slowly in these early stages. But the mean height of the Verticillium is reduced compared to the control in these treatments. I've summarized 10 of the 36 cultivars that we included in our test. These are the cane heights from the first study that we initiated in May. And for each cultivar, we show the non-inoculated control in yellow, the mean cane height, and the Verticillium inoculated plants in gray. Of course, we see a very highly significant effect of cultivar. Some cultivars are shorter, some are very tall, like Meeker or Munger. But the trend that we actually want to look at is the difference in the Verticillium treatment. And experiment-wide, as you can see in these 10 cultivars, there's a highly significant effect of Verticillium on mean cane height. The Verticillium plants are shorter. There are some cultivars that don't seem to fit that trend. You can see Tulamine and Willamette here, where the cane heights are similar to the controls, or the Verticillium ones are actually taller. And that could be an indication of resistance. It could be an indication that the disease progresses more slowly in these plants, or it could be part of the experimental error. The significance of Verticillium across the experiment is very highly significant at 0.01% significance. This is the same chart, but for the second study that we initiated in June 2013. And a very similar trend is occurring where there is a significant effect of Verticillium on reduction in cane height. The significance of Verticillium here is less. It's still significant at 2%. but it's less of a striking trend. And again, some cultivars are not showing that reduction. In this case, Meeker is not. And also Malahat, where the verticillium plants are slightly taller or very similar. The fact that the second test is showing less difference, less reduction in cane height for verticillium is an indication that the duration of this test is going to be very important for the validity of the results. So we have been prompted to prolong this test for that reason, and also for the reason that some researchers have speculated that some of the winter-associated damage that we see in black and red raspberry may be associated with verticillium, although that has not been experimentally shown yet. But we are planning to allow these plants to go through dormancy, accumulate chill, and then as they resume spring growth and into next summer, we want to monitor any effect of any verticillium interaction with cold temperatures and dormancy over that time. Then we will be able to take down the plants, take dry weights of the roots and canes, and also be able to examine the verticillium affected plants in their tissues to see if we can re-isolate verticillium from those tissues. And we hope to present those results next year. We want to thank our funders, the Crop Germplasm Committee and the WRRC, and also the people who provided such strong technical support for setup and maintenance of this experiment. I think we have some time for questions, is that right?
Yeah.
Okay.
And I just want to say one thing. One reason we're also carrying it an extra year is the positive control, the Munger that we inoculated in there, it really should have been hammered, and we didn't see as much damage as we would have expected from this round of inoculations on the screening. And it's a different system. We haven't worked in these size containers before. Sometimes the younger plants are a little more vigorous and resistant to infection, and that's also another reason why we decided to look at how symptoms develop next year, because I have seen other plants, the symptoms just get worse over time. So we're hoping to get a better idea of what is happening next summer. So, questions?
Any questions?
I am either completely awesome or we have obfuscated everybody.
All right.
All right.
Thanks.
Probably the former. Thanks, guys.